நான் தேடி வரைக்கும் படிக்கணும் இல்லையா? ஃபர்ஸ்ட் இயர் போயிட்டா தெரியாது. தேடி வரைக்கும் தெரியும். சரியா? எவ்வளோ ஃபர்ஸ்ட் பாருங்க. நீங்க வந்து இந்த நீங்க சொல்றப்பே சொல்லிருந்தோம் வந்துட்டு இந்த 3 நிமிஷம் புது உங்களுக்கு ஃபோன் போட்டு கொடுக்குறீங்க. ஆமா. நோட்டு வேணா கொடுப்பீங்களா? இதெல்லாம் வேண்டாம். மற்ற மற்ற வாங்கிக்கலாம் கொடுத்துருவேன். அவங்களுக்கு போட்டு ஹாஸ்டல் பையன் லிஸ்ட் ஹாஸ்டல் பையன் கரெக்டாக ஹாஸ்டல்னா ஹாஸ்டல் எழுத சொல்லுவாங்க. ஜெஸ்ட் தான் ஜெஸ்ட் சொல்லுவாங்க. ஹாஸ்டல்னா நம்ம நேர்ந்து இல்லையா? ஹாஸ்டல் இல்லை, ஹாஸ்டல் தான் எல்லாம் காமனாக அனுப்புற மாதிரி. யார் ஹாஸ்டல் கொடுக்கணும் என்ன போகணும்?
Hello. Can you all hear my voice?
Yes, bro, very clear.
Okay, thank you.
Happy morning, Professor Dean, sir.
Hear my voice? Morning.
Good morning.
Happy morning to all. This is Dr. Venkatraman, Dean, Science and Humanities.
Happy morning, sir.
Happy morning, sir. Dr. Suresh. Dr. Suresh.
Warm greetings to one and all join in today's session. One day international seminar on biotechnological advancement in health sciences. We, the Department of Biotechnology, SRM Institute of Science and Technology, Ramapuram, Faculty of Science and Humanities, Chennai, India, in association with Faculty of Medicine, University Sultan Zainab Abidin, Malaysia, and Laureate Institute of Pharmacy, Himachal Pradesh, Technical University, India, are jointly organizing this event today. To begin with today's session, now let's all rise for Tamil Thai வாழ்த்து.
சீராரும் மதனமென நிகழ்வரத கண்டமிதில் வெங்கனமும் அதில் சிறந்த திராவிடனல் திருநாடும் சிறு நிறை முதலும் தரித்தனரும் திலகமுனி அத்திலக வாசனை போல் அனைத்துள்ளதும்
I'd like to invite Dr. M. Kamaraj, convener and head Department of Biotechnology, Faculty of Science and Humanities, Ramapuram Institute of Science and Technology, to deliver the welcome address. Welcome you, sir.
Thank you, ma'am. I hope my audio is fine.
Yes, sir.
Okay, thank you. Very good morning to one and all. Joined here in this international conference in a virtual mode. On behalf of the organizing committee, I'm honored to welcome all the faculties, research scholars, and students who have gathered here for this international seminar.
Yes, sir.
This one-time international seminar on biotechnological advancement in health sciences, organized by Department of Biotechnology, Faculty of Science and Humanities, SRM Institute of Science and Technology, Ramapuram campus, in association with Faculty of Medicine, University Sultan Zainal Abidin, Malaysia, Laureate Institute of Pharmacy, Himachal Pradesh Technical University, India. With this introduction, I welcome our Dean, Dr. M. Venkatramanan, Dean of Research, SRM IST Ramapuram, and I welcome Dr. M.S. Ashawat, Director, Laureate Institute of Pharmacy, Himachal Pradesh, India. And I welcome our Professor Dr. Hami bin Muhammad Yusuf, Dean, Faculty of Medicine, UNISA, Malaysia. And I welcome Dr. Saravanan Dharmaraj, Associate Professor, Faculty of Medicine. Uh, in addition, I welcome Professor Dr. M.S. Ashawat, as I mentioned earlier, and Professor Dr. Kabil Kumar, Director, Samrat Prithviraj Chauhan College of Pharmacy, Kashipur, Uttarakhand, India, and Dr. Ahmad Najib, Associate Professor, National Product Chemistry, Faculty of Pharmacy, University Muslim, Indonesia. I welcome all the dignitaries who have joined in this session and who have given a consent to present their invited lectures in this international seminar. Welcome our Admin Vice Principal Dilipan Sir and Academic Vice Principal Saravanan Sir and all the research scholars, students, faculties who all have been joined in this international conference. I hope this seminar, international conference will be a opportunity, a platform to expand our knowledge and improve our understanding of the world and learning more about the new and existing research in the field of biotechnology. Once again, I welcome you all for this international conference seminar. Thank you.
Thank you, sir. Now, may I request Dr. Suresh, Assistant Professor, Department of Biotechnology, co-convener of this international seminar, to detail about the event. Welcome you, sir.
Good morning to everyone present here. I am truly happy and delighted to organize a seminar in association with Faculty of Medicine, UNISA, Malaysia. Our collaboration with UNISA began with our first international conference in 2021, and it was further solidified with a Memorandum of Understanding in 2023. This seminar represents a continuation of the Truthful Partnership. I would like to express my sincere gratitude to Professor Dr. Harmit Obadius, Dean Faculty of Medicine at UNISA, Malaysia. Working with you has been great pleasure. Your support has been invaluable, sir. I also extend my thanks to Professor Dr. Mahadev Rao, Faculty of Medicine, for his mentorship, which has been instrumental in bringing us together for this wonderful session. Thank you for your exceptional guidance, sir. A special mention goes to Professor Dr. M.S. Ashawat from the Laureate Institute of Pharmacy. Thank you, sir, for promptly accepting our invitation to collaborate on the seminar. Our cooperation has been tremendous and we deeply appreciate it. The seminar aims to provide a platform for researchers, academicians, and industry professionals to discuss the latest development and innovations in biotechnology, particularly in the context of health sciences. By bringing together experts from India, Malaysia, and Indonesia, the seminar seeks to enhance collaborative efforts and share diverse perspectives on biotechnological applications in health. Hence, the focus of the seminar is on how biotechnological advancements can contribute to solving critical health issues, improving patients' care, and developing new therapeutics. By involving participants from both academic institutions and the biotechnology industry, we aim to bridge the gap between the research and practical application, fostering partnerships that can lead to real-world solutions. The seminar will offer valuable insights and knowledge to students and emerging researchers, helping them understand current trends and future directions in biotechnological research and its impact on health sciences. In line with the objectives of the seminar, we have arranged 4 different intermittent talks today. By 9 o'clock, first invited talk by Dr. Saravanan A. Dharmaraj, Associate Professor, Faculty of Medicine, UNISA, Malaysia. Topic, functional analysis of obesity related genes selected by biometric analysis of gene expression data from mice fed high fat diet. Second invited talk, 9:45 a.m. Indian Standard Time, by Dr. M.S. Ashawat, Professor, Director and Principal, Laureate Institute of Pharmacy, Himachal Pradesh. Topic, biotechnology and personalized medicines. By 10 a.m. in Indian Standard Time, third invited talk by Dr. Kapil Kumar, Director, Samrat Purchi Chowdhury College of Pharmacy, Kashipur, Uttarakhand Technical University, Dehradun. Topic, the role of pharmaceutical sciences in addressing global health challenges. Last but not least, fourth invited talk around 11:15 a.m. Indian Standard Time by Dr. Hamad Najib, Associate Professor, Natural Product Chemistry, Faculty of Pharmacy, University Muslim Indonesia. Topic, Cardia myxa L, the potential plant as the anti-diabetic. These sessions will surely enhance and fulfill the aim of our seminar. I am pleased to announce that we have received a significant number of registrations with around 370 participants. Among them, 49% are UG, PG, and research scholars, 41% are academicians and scientists, and approximately 10% are industrial professionals. Participants have joined us from leading institutions and universities across India, Malaysia, USA, Indonesia, Pakistan, and so on, and several other countries. I wish the seminar every success in achieving its intended goals. Thank you all for your participation and contributions. Thank you.
Thank you, sir, for giving a detailed introduction about today's international seminar. I now welcome and request Major Dr. M. Venkatramanan, Dean, Faculty of Science and Humanities, SRM Institute of Science and Technology, Ramapuram, Chennai, India, to give away the special address. Welcome you, sir.
Thank you, Dr. Ansi, most respected chief guest for today's international one-day international workshop on biotechnological advancements in health sciences. Professor Dr. Harme bin Mohammed Yusuf, Dean, Faculty of Medicine, UNISA, Malaysia. Special guest for the workshop, Dr. M.S. Ashawat, Director, Laureate Institute of Pharmacy, Himachal Pradesh, India. Special invitees for giving lectures on various areas, Dr. Saravanan Dharmaraj, Professor Dr. Kapil Kumar, Dr. Ahmed Nazib, and the head of the Department of Biotechnology, Dr. Kamaraj, the convener, and the co-convener, Dr. Suresh. It is a great pleasure to be with you all today for this international workshop. I'm happy to see you all again since we met last year. I'm really happy to see you all today for the same workshop, which is an annual feature of SRM Ramapuram, Faculty of Science and Humanities, Ramapuram. Indeed, I should appreciate the Department of Biotechnology, the head of the department, the faculty members, and the organizers for having taken up this initiative and made it fruitful today, made it a success today by conducting it virtually. And this is the event held in association with the Faculty of Medicine, University Sultan Zainal Abidin, Malaysia, and Laureate Institute of Pharmacy, Himachal Pradesh Technical University, India. On behalf of, and it is also an MOU collaborated workshop, international workshop. On behalf of the Faculty of Science and Humanities,
Ramapuram. I extend a hearty welcome to today's guests, whom I have mentioned earlier, Dr. Professor Hamid bin Mohammed Yusuf, Dean, Faculty of Medicine, UNISA, Malaysia, and I'm delighted to note that this international seminar is a significant part of our MOU collaboration activity, as I told you earlier. This event marks another milestone in our journey towards achieving our goals in the field of health sciences and biotechnology. A special mention has to be made with regard to our distinguished guest, Dr. M.S. Ashawat, Director and Principal, Laureate Institute of Pharmacy, Biotechnological Advancement, Institute of Pharmacy. This conference, I would say that the biotechnological advancement plays a crucial role in health sciences, offering significant benefits. This seminar, featuring 4 eminent speakers from various higher education and research universities, surely will give you a very good insight and impact on the presentation. And I am sure the topics that have been chosen is really worth understanding, and it is relevant to our current situation with regard to dietetics, with regard to the gene expression from mice feed high-fat diet. So it's all mostly obesity, which is the order of the day, I should say. In fact, I am also a little obese, and I should learn from this conference what are the methods that will help us medicinally, biotechnologically, how to reduce the obesity. So it is the really a worth conducting international conference on the area, which is very important for the society as a whole. I, on behalf of the Faculty of Science and Humanities, I wish this international workshop a grand success, and I'm sure that the seminar will provide the younger generation, academicians, researchers, and industry professionals with fruitful discussions and valuable knowledge in their respective field of focus. I am really once again thank the organizers for having given me this opportunity to address you all. And also I welcome all the participants, both national as well as international, for this workshop. I wish once again this workshop a grand success. Thank you for this opportunity. Thank you. God bless you all. Jai Hind.
Thank you, sir. Thank you for your blessings and the special address delivered. I welcome and request Dr. M.S. Ashrawat, Director cum Principal, Laureate, Institute of Pharmacy, Himachal Pradesh, India, to deliver the special address now. Welcome you, sir.
Very good morning, one and all. Am I audible, sir? Good morning from the land of Lord Himachal Pradesh, Jwalaji. I am Dr. M.S. Ashawat. I am very much thankful to the UNISJA University, SRM Institute of Science and Technology, Department of Biotechnology, and special thanks to Professor Rao, who is conveying such a international webinar in the leadership of convener Kamraj sir, as well as co-convener Dr. S. Suresh. I convey my thanks and pay regards to Dr. Sarvan Dharmaraj sir for sparing the time to deliver his expertise on functional analysis of obesity and related genes. So certainly, sir, our audience will be benefited with your expertise of the field. I am much more thankful to Kapil Kumar sir, Samrat Prithviraj Chauhan College of Pharmacy, as well as Ahmed Najib sir from the Natural Product Chemistry Department of Pharmacy. I am really thankful to all the audience as well as administrator of this Sultan Jainal Abid University, Malaysia, for organizing such a wonderful international webinar. And I, on behalf of Laureate Institute of Pharmacy, welcome all the speaker on this platform, audience, and I wish you all the grand wishes from our institute side. Thank you. Thank you very much, sir.
Thank you, sir. Thank you for the special address delivered. I now request Dr. Vinod Kumar, Assistant Professor, Department of Biotechnology, Faculty of Science and Humanities, SRM Institute of Science and Technology, Ramapuram, to introduce today's chief guest. Welcome, you, sir.
Good morning, everyone. It's my honor to give an introduction to such a wonderful, experienced person, Professor Parmi bin Mohammed Yusuf. He is currently as a Dean, Faculty of Medicine, UNISA, Malaysia. He did his medicine degrees from the University Kebansan, Malaysia. He have number of years of experience. He started as an house officer, and then he served as a medical officer, lecturer, head of the department, Deputy Dean Academic, and reached to the level of Dean Faculty of Medicine. And his area of interest includes clinical diagnosis and treatment. He has published number of papers in reputed national and international journals, and also given lectures in various conferences, and given number of invited lectures as well. In addition to publication in journals, he also published number of teaching models to help students to learn the concepts, as well as he published several academic books, and for the benefit of the public, he has published several popular articles in the magazines. And he also received funding as a grant from several funding agencies at national as well as international level. He received fund in the capacity of principal investigation, co-investigation, and co-researcher as well. He has also supervised number of master students in medicine, in addition to several graduate students. He received several awards and accolades from number of bodies, and he has served as editorial board member in several journals, and he has also become a member as well as a consultant in various boards and committees. And he has a number of years of experience, around 25 years of experience in academic research and administration. So with this, I would like to welcome again Professor for joining in this particular one-day international seminar as a chief guest. Thank you so much for the opportunity given.
Thank you, sir. I now invite Professor Dr. Hamid bin Mohammed Yusuf, Dean, Faculty of Medicine, UNISA, Malaysia, to deliver the inaugural address. Welcome, you, sir.
Okay. Thank you very much, Madam Chairperson. Assalamualaikum warahmatullahi wabarakatuh, and very good morning to everyone. Ladies and gentlemen, distinguished guests, esteemed colleagues, and dear participants, especially it is my honor and privilege to welcome you to this international seminar on the unveiling biotechnological frontiers in the medical science. I think this event is represent a significant milestone in our collective journey towards advancing health sciences through the biotechnological innovation. I extend a warm welcome to our distinguished collaborators from UNISA, from SRM Institute of Science and Technology, Ramapuram, India, and Laureate Institute of Pharmacy, Himachal Pradesh Technical University. I think your presence here today underscore the importance of the international cooperation in driving the scientific progress. I am particularly delighted to acknowledge the presence of Dr. M. Kamaraj, Associate Professor and Head of Department of Biotechnology, and Dr. Suresh S., our co-convener and Assistant Professor in SRM Institute. And your dedication has been instrumental in organizing this seminar. And I also honored to have Dr. Venkatamaran, Dean of the Faculty of Science and Humanities at SRM, and Dr. Mahendra Singh Ashwat, Director and Principal of the Laureate Institute of Pharmacy, with us today. So your insight and leadership are invaluable to our endeavor. I think today's seminar is very important. It brings together a remarkable assembly of minds to explore the groundbreaking stride and transformative possibilities that biotechnology offer to the medical sciences. I hope all the participants from India, Malaysia, Indonesia, Pakistan can come together. It is a testament to what can be achieved through collaboration and shared vision. We are witnessing an era where the biotechnology is revolutionizing healthcare. From the precision of genomes editing tool like CRISPR-Cas9 to the promising horizon of personalized medicine. And I think this integration of artificial intelligence in medical research is a potential matters. And these advances are not just scientific milestones, but they are represent hope for 1 million around the world. A lot of diseases previously incurable now become curable because of the understandings of the genetics and bioinformatics. However, with the great power come with the great responsibilities. As we push the boundaries of what is possible, remember, we must remain steadfast in our commitment to ethical practice and equitable healthcare. Remember our duty to ensure that these advancements benefit all humanities. It must be guided by the principle of fairness and justice. And I'm confident that the discussion and knowledge shared today and tomorrow will pave the way for future collaboration, strengthening our collection effort in research and innovation. And I hope this seminar be a catalyst for new ideas, a partnership, and groundbreaking discoveries that will propel us into a future where the benefit of the biotechnological progress are fully realized. And again, I offer my heartfelt congratulations to the organizer and speaker for their dedication and hard work in making this event possible. Let us all actively engage in the discourse, share our insight, and work together towards the future where the biotechnology transform medical sciences for the betterment of all. Once again, I hope that all the input today and tomorrow will bring us to a betterment in healthcare. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor. Thank you for the inaugural address you have delivered. I now invite Dr. G. Priya, Assistant Professor, Department of Biotechnology, and co-convener of the seminar to deliver the vote of thanks. Welcome, ma'am.
Good morning to one and all present here. On behalf of Department of Biotechnology, SRM Institute of Science and Technology, Ramapuram, I am glad to be standing here to express my vote of thanks. I wholeheartedly thank to today's remarkable chief guest, Dr. Professor Hami bin Mohammed Yusuf, Dean, Faculty of Medicine, UNISA, Malaysia, for gracing this international seminar on biotechnological advancements in health sciences with his valuable knowledge and ideas. My sincere thanks to Professor Dr. Mahendra Singh Ashawat, Director and Principal, Laureate Institute of Pharmacy, Himachal Pradesh, for his special address of our seminar. I would like to thank our invited speakers of today's international seminar, Dr. Saravanan Dharmaraj, Associate Professor, Faculty of Medicine, NISA, Malaysia, Professor Dr. Mahendra Singh Azrawat, Director cum Principal, Laureate Institute of Pharmacy, Himachal Pradesh, Professor Dr. Kapil Kumar, Director, Samrat Prithvi Chauhan, College of Pharmacy, Kashipur, Uttarakhand, and Dr. Ahmed Najib, Associate Professor, Natural Product Chemistry, Faculty of Pharmacy, Universitas Muslim Indonesia, for their acceptance. I would take this opportunity to place on record our hearty thanks to our management for their constant support and encouragement to organizing this international seminar. A special thanks to Dr. M. Kamaraj, Associate Professor and Head of the Biotech, for being the catalyst that stimulate us to do our best and standing as a pillar of strength. I would also like to mention a special thanks to Dr. S. Suresh, Associate Professor and co-convener, for his guidance and continuous motivation for successful conduction of this seminar. My hearty thanks to all my colleagues for their teamwork and effort to make this seminar a successful one. A huge thanks to participants who are all making this event more memorable one. Your enthusiastic participation and discussions will motivate us to conduct this type of remarkable events in all time. So once again, my thanks and wishes to all participants and expecting the same in all our future programs. I'm sure this seminar will be in fruitful manner to gain enormous knowledge and ideas on all recent advancement of biotechnology, mainly in health sciences. Finally, I would like to thank each and every one of you for being here and making this day a wonderful one. Thank you all.
Thank you, Priya ma'am. Now, let's begin with the session of the event. For the first session, I'd like to introduce the chief guest of the first session. So with pleasure, let me introduce Dr. Saravanan Dharmaraj, the invited speaker of session 1. Sir is presently working as Associate Professor, Faculty of Medicine in University of Sultan Zainal Abidin of Malaysia. He pursued and completed his PhD in pharmaceutical chemistry in the year 2008 from University Sains, Malaysia. His area of expertise includes chemometrics, molecular pharmacology, traditional medicine, and natural products. He is a professional member of Malaysian Society of Pharmacy and Pharmacology and American Association for Advancement of Sciences. He has participated in nearly 8 conferences and training programs at international and national level and shared his research knowledge. He has published about 24 research articles in various reputed journals. He has 3 book chapters to his credit published till now. He has received Excellent Service Award of the Year 2013 by UNISA. So with this short introduction, I'd like to welcome you, sir, for this session and request you to preside over the session, sir. Welcome you, sir.
Thank you.
Dr. Saravanan, sir?
Can you hear me?
Yes, sir, it's audible, sir.
I think I will start my talk. I can share the screen.
Yes, sir, please. Yes, sir, your screen is visible, sir.
Okay, thanks. A very good morning to all the participants. I would like to thank for the opportunity to present my research, and I would like for opportunity to collaborate with you all in the future. This session is more of a knowledge sharing opportunity, of which I believe my knowledge in this area is very limited. And in this session, I'm going to talk about the functional analysis of obesity-related genes selected by chemometric analysis. This is actually a in silico technique. I'm using data that is available. And in this talk, maybe I might initially disappoint some of them by telling that I'm not going to talk too much about obesity, its mechanism of action, how it causes the pathology and all that. I'm sure others like Dr. Rao and the participants are very aware of these things. What here I'm going to talk about is the analysis. The main reason is because obesity, basically it arises from a complex interaction of many factors. of which genetic predisposition, lifestyle, and others play key roles. If you see from the point of genetic this thing, there are various genes which have been implicated in obesity. So the goal of my work is to understand what is the role of some of these genes and how do they work together with other molecules. For example, DNA produces RNA, RNA produces acid amino, okay? And this process is a bit complicated. Other molecules have a role to play. So what you want to have here is a better understanding of the correlation between genes and functional phenotypes of an organism at the functional level. I'm sure you all have heard of artificial intelligence, machine learning, and these things. For example, few years back, the world champion in the game of Go was beaten by artificial system known as AlphaGo. The main thing about AlphaGo is those who programmed it, they did not give much knowledge to the software or the program. They just gave it the rule to win, okay, and let it develop itself. Okay, so it played games with itself, and then finally, okay, it learned, and then its knowledge was good enough to beat the world champion. So in like that kind of approach, okay, what it does is it learns by itself, and also it does not know what are the starting rules. So in functional genomics, like in obesity also, I believe if you really want to understand the mechanism, we have to start from nothing, analyze the data from an unbiased perspective. And this is what I have done. Okay, as I mentioned just now, okay, based on the central dogma, okay, heritable information flows from DNA to RNA to proteins and then to phenotypic traits. Okay, the conventional understanding was DNA produces mRNA and then translation of it produces protein. But recent understanding of molecular biology has altered this view. For example, as early as, recent as 2012, okay, it was shown that DNA, okay, produces mRNA, and this is translated to protein. Okay, and the regulatory RNAs, for example, microRNA, siRNA, okay, pi RNAs have a role. Okay, if you compare this with now, okay, there are other molecules, there are long coding mRNA, long coding RNAs, they are circular RNAs. So there's a big family of molecules which are related, and it's difficult to understand their role. So one way, for example, if we see the information flows from the genome to epigenome, from that to the transcriptome or mRNA, and then in the proteome level, it produces the proteins, and then at the metabolome level, okay, the metabolites are produced, and these have a role, for example, in like metabolic syndrome, of which obesity is one of it. Okay, and you can see, for example, okay, the proteins, okay, they can bind through the transcription factor binding sites to DNA and influence the thing. At the level of epigenome, there can be reactions like methylation of DNA, which will have effect on the gene, or at the level of transcriptome, alternative splicing can interrupt the protein distinct production. And here also, I would like to mention about at the metabolome level, okay, there are many techniques which detect the different molecules and things like that, but that approach is not that feasible, okay? Techniques such as metabolite fingerprinting, which use pattern recognition techniques to obtain the overall pattern without identification of specific molecules, are, I believe, more of the future thing to do. Here, in this talk, I'll be mentioning a bit about the microRNAs, which are small 19 to 22 nucleotide long molecules, which play a critical role in the regulation of gene expression by targeting mRNA for degradation or by suppressing the translation. How it is produced, okay, and its role is shown in this slide. I will not go much into detail. Most of the information given in the slides are available with me, and you can contact me for them or for like further discussion. Circular RNA, although it was discovered about 40 years back, okay, now it has a prominent role, has been discovered, and more work is being done with it. So in this thing, to understand all these things, what I'm suggesting is data reuse and integrated data analysis. There's a lot of data, and the idea is to use existing data to make new discoveries. And in the methods, okay, which I discussed, I will show how this can be done. You all have heard about data a lot, okay, but the important thing is the high quality data is the most important thing. Because if you start with data which is not of good quality, the results will not be good. Available data repositories for these studies are from the Gene Expression Omnibus, or like the Array Express Functional Genomics data. The websites for this are shown. The data can be obtained from this database or this one, and we can be used for further analysis. As I mentioned just now, the approach I'm using is chemometrics. Basically, it is a discipline at the intersection of chemistry, statistics, and data analysis. It applies statistical methods and mathematical models to interpret chemical data and extract the meaningful information from these data sets. Other related fields are data science and machine learning, which can be considered, all these can be considered subsets of artificial intelligence. Here I would like to mention about data science. Data science is an interdisciplinary field that combines math, statistics, programming, and subject matter expertise to extract insights from the data. It involves techniques such as data mining, cleaning, visualization, machine learning, and statistical analysis to gain knowledge from the data. subject matter expertise is important. For example, doctors might have, or those in the medical practice might have some knowledge about some disease, for example, some genes which could be important. Okay, so this information could be integrated into the study, as I shall mention later. In other fields, like in herbal research, which I'm also doing, sometimes we measure FTR spectra and things like that. Okay, and what we know, certain regions of the spectra are not useful. They are to be discarded. So this subject matter expertise has to be used in the data science to make the analysis better. Machine learning and deep learning, these are also techniques, related techniques. Okay, the difference is slight in some of them. Deep learning basically uses a neural network, and deep learning can be considered as a more complex system. The perceptron is a basic unit of a neural network, and neural network usually has about 2 hidden layers, whereas deep learning typically employs more than 4 hidden layers of neurons for complex learning tasks. Deep learning, one of the disadvantage of it is that it is an opaque system where you can't find the underlying reasons which are separating the samples and things like that. This slide shows the relation between all these fields. Now I come to the search space. Search space is important. For example, in genetic algorithm, it is able to explore a vast space and produce better results. Data used in the study. Okay, the data used in the study is from the study of Doe et al. 2011. Okay, and this study observed the effect of high-fat diets on metabolism in the liver. Okay, this data is available, and the number of samples about 48, and the data used is of the normalized signals from the Illumina probes. Other data used in this study is the microRNA database, which shows interactions between microRNA from humans and target genes from human. This plot shows how principal components are calculated. This is the important basis for the one of the analysis. As you can see from the diagrams, okay, the principal component 1 is where, okay, most of the distribution of the data is the most, whereas the principal component 2 is the one chosen, which is orthogonal to the PC1 and contains the next level of total variance. For this analysis, okay, it was done using RStudio. The language, programming language was R, and the top 50 gene variables with the highest loading in the principal component 1 were chosen for further functional analysis, and the top 30 variables were used for classification. The reason for using lesser samples for classification is because if you use the total 50, the classification results are not that good. So it's better to limit the variables used for classification, whereas to get the functional annotation, the functional meaning, how the genes function, a bigger choice of genes are necessary. This shows a decision tree, and a random forest is basically a collection of many different trees. And here, the library of random forest in RStudio was used. Some of the parameters for the random forest analysis was mtry, which was set to 120. This is usually for this parameter, we use the square root of the total number of variables, okay, around there. So the total number of variables in this thing was there, and this was the was 15,000, and 120 was quite close to the square root of it. The number of trees is also another important parameter and was set to 1,000. And the selection of genes were done based on decrease in accuracy. The other method for calculating genes are based on Gini, Gini, okay, but decrease in accuracy produces better results. The method for genetic algorithm, okay, where we initialize population and then select individuals for mating, and then okay, a certain criteria is used, okay, to select the offsprings, okay, and here in the study, the method of Kemsley 98 as adopted in Dharma 2021 was used. Okay, the loadings for canonical variates for 3 dimensions were calculated by running the algorithm for 400 generations. Okay, the number of chromosomes were 100, and the variables with the highest loadings in the first dimension were chosen. Classification. Once you've chosen the variables, it is necessary to classify them. Okay, and here, support vector machine with linear kernel as well as k nearest neighbor were used to classify the samples. The library E1071 in RStudio was used, and the classification ability of the variables chosen by the 3 methods were considered as the average of the KNN and SVM technique. Functional annotation of the chosen genes relating to biological processes, cellular components, as well as molecular functions were studied using David. And the important protein-protein interactions were studied using string database, and the plots were analyzed using cytoscape. The biological meaning of the chosen mice genes were extrapolated to regulatory aspect of associated human genes by human microRNA using the site-target linker. These are the hardware and the software used. I just wanted to show you this slide. This I would call as a takeaway for one of the takeaways for this lecture. If you see here, okay, in the famous book, the author has mentioned, okay, the gene name, gene symbols are like complicated. You can't find meaning in them, okay, and that is true. Okay, what you need to know here is, okay, take difference between the human gene, okay, and the mouse genes. Okay, for example, the genes for humans are use the full name, use the capital this thing, whereas the other one does not use the capital this thing. But what you need to take note is the gene symbol. Okay, the gene symbol uses italics for humans as well as for the mouse or other species. Okay, but this capitalized for the human. Now I'll mention about the differential genes between the high-fat diet and the normal diet. The principal component showed that the principal component analysis showed that the principal component 1 contributed 27.3% of the overall variance, whereas PC2 was responsible for the remaining 17.6%. And a total of 21 principal components were required to achieve the cumulative proportion of variance of 90%. These are important parameters usually you report, okay? Because usually in some methods, the 90% will be achieved by using few principal components. And the variables for further analysis were chosen based on the highest loading for the PC1. The selection of genes by RF, random forest base, was based on the mean decrease in accuracy, and the selection of genes for GA based on highest loading for the canonical variants for the first dimension. The top 6 genes out of 50 chosen for this are shown, and the ones highlighted, okay, are the ones that have some role or major role in the obesity. Okay, the percentage of correct classification of the test samples was 100% for both PCA as well as random forest, whereas genetic algorithm obtained lower percentage of correction. Okay, this was based on using the two methods of KNN and SVM. Whereas if other techniques are also tried, okay, PCA obtains a higher value compared to random forest. Functional annotation clustering, okay, showed that the variables for principal components were more related to glutathione transferase activity, glutathione metabolic process, xenobiotic catalytic process, xenobiotic metabolic process. Okay, generally it can be said as general metabolic processes. This shows the protein-protein interactions. As you can see, okay, these things are often complicated, and what you are, and for if you choose more genes, it will become more complex and it's hard to understand. Okay, what is important here to detect small modules in them which have some role. For example, okay, if you use the module analysis of M-code, you find that one of the networks, okay, is involved in glutathione transferase activity as well as glutathione metabolic process. Okay, and you can also see, okay, what are the hub genes. Okay, the hub genes are important for therapeutic or diagnostic purposes. Okay, and these are the ones that are identified by this method. Okay, the human microRNA interaction with the single protein, basically the human microRNA, okay, all these ones can influence the MGST3 protein production. Okay, and this is just for single protein is shown. Okay, for the overall network, it is much more complex. I will explain the significance of, okay, once of this thing later. Okay, one of the proteins you can see in mice hepatocytes, okay, CDA is necessary for SREBP1 activated lipid accumulation and triglyceride storage. Okay. For the functional clustering from random forest selected genes, you can see that here, okay, it identifies processes related to inflammatory response as well as processes related to cholesterol metabolic process, as well as sterol biosynthetic processes which are important or related to obesity. I just want to mention that 3 of the genes identified by PCA were also identified by random forest. And here you can see from the functional module analysis, okay, that one of the modules, okay, the group of proteins have a role in cholesterol metabolic process as well as cholesterol biosynthetic process. Functional annotation clustering for genetic algorithm selected genes. You see that the number of functions for these genes are less, okay, and these are quite related to process of like spermatogenesis, cell differentiation, and multicellular development. In this, okay, no functional module clusters were identified, and also basically the hub genes had less interactions compared to the previous ones. Okay, the 2 hub genes identified were like KIF23, EF1D, and NME8. Okay, and also you need just would like you to note that another protein identified, okay, PLB2, okay, is associated with risk of breast cancer as well as also downregulated in hepatocellular carcinoma. This slide shows the microRNA RNA interaction, okay, for the overall genes, okay, selected by genetic algorithm. Okay, this for the network for this is much less complicated, and you can also see, okay, for example, NME8 could be a potential therapeutic target, okay, because, okay, its production, okay, is not influenced much by human microRNA. This shows what happens when you combine the genes from PCA and RF. Okay, and what you can see, okay, you get a much wider interaction. Okay, the process involved becomes more complicated, but you can also see, okay, that the module proteins, okay, are extended, okay, and there are more interactions. Okay, so this is also good in sometimes when you want to study more interactions. Okay, it is good to combine the proteins selected by different methods. Okay, the findings from these studies, okay, are the different selection methods identify genes with slightly different functional terms. And these are important because if you take the microarray data, you will see a lot of genes. When you study the expression studies, a lot of them have upregulated and downregulated expression. Okay, you find a lot of genes, but you can't understand, okay, by itself, okay, which are the important ones. Whereas like this, you can have choose genes, okay, based on different functional modalities and can analyze them further. Also, you see that the genes selected by RF, okay, had higher classification ability, okay, and one of its protein modules had a role in cholesterol metabolic process. Its genes also showed inflammatory function and cholesterol metabolic process in functional analysis. Okay, the gene selected by PCA had a role in glutathione metabolic process. The gene selected by genetic algorithm had less protein-protein interactions as well as functional annotation. Okay, but this study shows that they may have potential for further study. Usually, genetic algorithm is a technique which is optimization technique which is very good. It can find, for example, what are the variables, okay, between the 11,000 genes, which are the ones that produce the best interaction, okay, and this technique also often in other, when using other data, has good classification ability. Limitations of this study, okay, number of samples was small, okay, and the analysis was limited to 15,000 out of about 20,000 genes in the data, okay. The reason for this, okay, one of it is because of the computation cost. Okay, some of the analysis takes too long if you're using more genes. Also, another limitation is, okay, that the genetic algorithm can be further optimized to obtain better solutions. Future study, okay, the method is to be expanded to include, for example, like N colony optimization. Okay, this work is in process. Okay, but what I find from using N colony optimization, that it narrows down to a very specific region. Okay, it narrows down to a very specific region, and I'm not sure, okay, whether the this is due because, okay, to the reason that maybe the genes, those particular genes are very good for classification as well as functional testing, or is there some problem in the optimization? Okay, variable selection by PCA, RF, and GA, okay, has been used for metabolite fingerprinting of local Malaysian herb. Okay, and it can also be used for other data, other domains. Okay, even domains, all these selection methods can be used for domains in social science, etc. Okay, and the data from study of single cell expression is to be compared. Okay, this will be good when you compare data like the existing one with data from single cell expression studies. These are the references, and thank you. These are the papers.
Thank you. Thank you, Dr. Saranan, sir, giving insights about the gene data sets and for single cell expressions, metabolite fingerprints, and interactions of proteins. Also, protein models and its role in glutathione, certain limitations, and also about the genetic algorithm, which can be used for a better solution identification. Thank you for your presentation, sir. So the second session, may I invite Dr. R. Vinod Kumar, sir, to introduce the resource person of the second session. Welcome, sir.
A very good morning, everyone. It's such a wonderful lecture you have listened in the session one. So I would like to introduce the today's speaker for session 2, Professor M.S. Ashawat. He is currently the director cum principal at Laureate Institute of Pharmacy, which is affiliated to Himachal Pradesh Technical University Research Center in Himachal Pradesh, India. He holds a postgraduate degree in pharmaceutics and began teaching in way back in 1999. Dr. Ashawat's career spans various prestigious institutions where he introduced innovative teaching methods. He has received accolades for his contribution to academics and is recognized for his diverse research institutes include intradermal depots and herbal cosmetics. He has authored several books and he wrote chapters in various edited books and published numerous research papers and also guided several graduate students for several universities. He has also held leadership roles in professional organizations, showcasing his commitment to education and research and development. He has published several books and chapters, in addition to 85 research and review papers in international and national reputed journals, and presented more than 100 papers in conferences and seminars and invited lectures and international reputed institutions. Dr. Ashawat has served as State Vice President of APTI and presently working as National Coordinator of APTI, assuming the overall responsibility of a devoted educator covering professional development, member of various committees of state technical universities concerned with affiliation and staff selection, and Himachal Pradesh Technical University affiliation committees. With this, I'm heartfully welcome Dr. Ashawat, Professor Ashawat, to give his lecture in this session too. Thank you so much. I welcome you, sir.
Yes, good morning. Am I audible?
Yes, sir, you are audible, sir.
Very good morning, all the ladies and gentlemen, all the participants. It's my distinct honor to welcome you to this international conference on biotechnology and personalized medicine. This gathering brings together the brightest mind in the field of the biotechnology and life science, all the researcher, practitioner, and our all the speaker from the distinguished and their expertise field. Our discussion over the day will focus on the latest advancements as our conference is titled in the biotechnology, and how they are revolutionizing the personalized medicine. I will little bit discuss on this pivotal moment. So together we will explore the groundbreaking research, as our previous speaker also highlighted on the genomics aspect. So in the same view, I hope slide is sharing. My screen is visible to all of you.
No, sir.
The screen is not visible. I shared the screen. Now it's visible, sir?
Yes, sir. It is visible, sir. You can carry on, sir. Thank you.
So, as we all know, I'm from the pharmacy research background. So, I would like to tell you that about the precision medicine, personalized medicine, which is rapidly expanding, as we all know, the field of practice that uses individual genetic profile to guide decision about the prevention, diagnosis, and treatment of the disease. Although the concept of personalized medicine, it's not a new term. Personalized medicine started, means the idea click in 1890 when the tetanus vaccines were introduced, but this term coined in 1990 following the advancement in DNA sequencing technology. As we all know, around 135 years ago, 1899, the cell and its omics and its organelles were identified by the researcher. So from that beginning, DNA sequencing technology also comes in the books, and it pretty much remained just a concept, and it was readily applied. So since then, continued advances have been generated with enormous amount of new information. The discovery of the gene proteins and the pathway has enabled studied into the genetics. If we talk about the bioscience beginning of the technology, it's a story that began in 1890 when the biological medicines first discovered. And from this beginning, we were capable to reach to get the antitoxins and to find out the treatment for the diphtheria and tetanus, which was a very dreadful disease that time. And so many lives have been saved. Another, all cancer patient which do not have the same prognosis. So each patient require a personalized treatment plan and not the same treatment as others. The patient tumors type will be profiled, and cancerous genomics has the potential to make the personalized therapy. In reality, as we know, the physics also play a vital role when the X-rays comes into the research. Then we were able to find out the area of the cell proliferation to get the proliferation size of the tumor also. And very earlier, my previous speaker, Professor Dharamraj sir, he explicitly elaborate on the functional genomics as well as glutathione transferase. In his previous slide, you have seen. So glutathione play also a significant role in personalized medicine due to their involvement in the detoxification process and their influence on the drug metabolism also. So here are some key points regarding the glutathione transferase also. In the drug metabolism, glutathione are enzymes that catalyze the conjugation of glutathione to various electrophilic compound and adding to their detoxification. In the same in the pharmacogenomics also, such variation in the glutathione transferase enzyme genes can affect an individual's responses to the medication. So, so many aspects are associated. Gene polymorphism is also there. We will discuss one by one how gene polymorphism will affect the disease treatment. Now you will see that beginning when we started the tailoring of the gene, because tailoring of the gene was the first step to learn about the individual DNA cell and which can create the proteins that fight diseases. So by keeping such thing in the mind, we were enough capable to evolve and develop the molecular diagnosis and genomic analysis, which increase the possibility of an even more understanding and interpretation of the human genome and exome also. So personalized medicine approaches to the clinical care and symptom medicine, symptom biology, all these are the terms correlated. Now you see the famous example of the our insulin. So type 1 इंसुलिन टाइप 1 जो डायबिटीज के बारे में आप जानते हैं, यूएस अप्रूव्ड दिस टाइप 1 diabetes treatment by the insulin. It was approved in 1982. It is expected that future personalized healthcare will also benefit from the combination of such personal genomic information with its longitudinal global monitoring of the molecular components. So you will see that there is a lot of research were simultaneously going on. But in a nutshell, we can find that if we are enough capable to find out the nature of the cell and its genome, because cell is obviously there is role of the genes and DNAs. So how we can alter the DNAs? There is a progressive scientific research has been conducted, and in year 2006, the Europe approved the first. It's one kind of biosimilar in America. So biosimilar compounds was a milestone in the personalized medicine in the 21st century, where to increase the treatment option by using the biologicals. And this is the time we will absolute the antibiotics from the market, and very soon biologicals will come forward for the treatment. As the concept of the personalized medicine, it's revolutionized the healthcare system right now, because it's wholly depends on the genetic profiles and the lifestyle and environment. So if we are capable to find out the specific gene of the person, we can find out the treatment to treat that particular gene. So personalized medicine is we can understand like that it's a tailoring kind of treatment of for the individual. So by using the different steps one by one, we can make the tailoring treatment for the individual. So there is a treatment plan for the pharmacogenomics. Targeted therapy, by reaching the targeted therapy, we can soon to prevent the disease by detecting its symptoms. And now I think it is very much clear. So there is certainly role of different lifestyle also. I will put some example how lifestyle will affect the patient treatment, and how data of the patient of the same disease will affect the treatment, even dose of the drug. You see, understand by this example, once we think more about the that one size does not fit all. Right now our treatment mechanism is like that कि हम एक ही दवा को we are using the same medicine to the all patient. So it's not possible at all. Instead, the healthcare provider recognize that each patient is unique with a distinct need, with its individual preferences and its circumstances. What perspective says that one patient is an elderly individual with an history of heart disease, while the other young is athletics, and their treatment plan cannot be identical. Say, for example, the elderly patient may require the comprehensive cardiac workout, including the stress test and echocardiogram. A doctor prescribed the test, whereas the athletics with the same pain may targeted focus on the musculoskeletal kind of issues. So there is a need to find out the depth of the patient symptoms also, and certainly there is role of the pathophysiology and individual test and diagnosis. So diagnosis affect much more impact on the personalized medicine. It's you can see that any patient with primary care physician evaluating the patient with chronic fatigueness, and instead of ordering a generic battery of the test, they must look into the account of patient occupation also. Sometimes the fatigueness may be due to some biological or hormonal imbalances, but sometimes it may be due to lifestyle, just like a night shift worker. So sleep pattern and mental health also affect the same thing. So the physician must know and prioritize to find out the sleep pattern of the individual patient, and Then they can assess the function of hormones like thyroid screening test and all, because all these affect the depression. So this personalized approach not only expedite accurate diagnosis, but also minimize sometime unnecessarily test. Another, we can discuss the example as far as concerned with the personalized medicine, which works for the one patient may not be suited for another. So you can see that there is a lot of genetic mutations occur during the cancer cell. So while the chemotherapy remains standard treatment, and some patient may carry the specific gene mutation. So for the specific gene mutation treatment, the tailoring therapy is required. The patient with the BRCA1 mutation, if there, so might benefited from the targeted therapy like PAR polymerase inhibitors, because such polymerase inhibitors may sometimes helpful, and they are safer. There is certainly the adverse effect of each and every drug. So you can see the example of the diabetic patient also. There is also need to tailor the treatment, just like you just see the example, the patient armed with the informative all the kind of information, but choosing between the metformin and sodium glucose co-transporter 2 inhibitor. So initially the therapy in the most patient with type 2 diabetes should begin with diet and body weight reduction, exercise, and metformin in absence of the contraindications. So there is alternatives are there, but there is certainly need to think doctor and pharmacist must, as well as patient must sit together, and then they can find the uh, better solution with the same drug, but different doses. Now, I'm just considering the example for the cancer genomics, as such cancer genomics help us to understand the basic cancer biology. There is again requirement of the personalized treatment, because different cell mutations require the different kind of drug treatment. It's not uniform for the all. As Barack Obama in 2015, they launched this precision medicine treatment. So precision medicine, sometimes known as personalized medicine, and the tailoring medical treatment to the specific characteristic for the each patient. It does not literally mean the creation of the drug and medical devices that are unique. You can see here the young children is there, or older adult is there, right? So there might be some immunological challenges for each age group. So subpopulation that are uniquely and disproportionately susceptible to the particular disease, and some may not be. As during the corona, it is proven. Now, in the biotechnological intervention, once we plan for the personalized medicine, there is several terms should keep in the mind, just like genomics. So by keeping the genomics pattern of the particular patient, the entire set of the genetic instruction found in the cell. So we must know about the genomics. We must know about the pharmacogenomics, where the pharmacology is concerned, with using the DNA and several amino acid sequences. Then pharmacogenetics also. So in the pharmacogenetic field, there is also, means there is lot of research is going on right now. to individualize the patient differentiation response to the particular drug. So these are the field where the scope for the personalized medicine is there. Then diagnostic paradigm shift and changing. So the trial and error dose existing in the market, but there is need to correct the medication for the patient, and various non-steroidal as well as anti-inflammatory agents for the treatment of osteoarthritis is there. Most effective agent may not always be utilized as the first increasing the cost of treatment, and certainly exposing to the patient to the adverse effect also. So there is changing in the medical diagnostic paradigm. Changing in the medical diagnostic paradigm, we must think about how do genes influences the medication responses. As we know, Human Genome Project has estimated thousands of the genes and genetic variation, and right now due to the changes in the gene, the polymorphism is occur, and due to the polymorphism there is also changes in the treatment. I'm just putting one example of the single nucleotide polymorphism. SNP is playing the vital role as a tool in the personalized medicine, because such type of single nucleotide polymorphism are the most common type of genetic variation. There is changes in the sequence of the amino acid sequence, because adenine, guanine, thymine, and cytosines are in the particular sequence, but upper DNA molecules sometimes differ from the lower DNA molecule at the single base pair. So such single nucleotide polymorphism expression may also modify the drug therapeutics responses as well as adverse effect incidences. So we must keep such type of issues during the drug design, how such single nucleotide polymorphism affect the drug therapeutics responses. Now, in the personalized medicine, such type of natural variation, just like a DNA polymorphism. So there is a lot of factors which are involved and contribute to disease risk in any individual genetics. And there is a work in concern with the other factor also, and genetic variation, which are variation to increase the disease risk, because due to the genetic changes, genetic changes due to the external environmental factor also affect the individual risk for the disease. And variation in DNA can lead to the difference in the pharmacodynamics as well as pharmacokinetic in the individual patient with a particular drug. So such kind of genetic variation can alter the pharmacokinetic as well as dynamics of the drug, and potentially affect both drug efficacy as well as toxicity. It will alter not only affect, it affect the adversity also as well as toxicity. So many evidences indicates that the genetic factor can account for an estimated to 95% of the drug metabolism and its responses. Now, there is technology, DNA microarray technology. So by using such kind of microarray technology, we can find out about the mRNA messenger RNA, and which is the body's template for the creating proteins. So such DNA microarray array technology and tool is used to determine whether the DNA from particular individual contain mutation genes or not. So the chip consists of a small glass plate encased in the plastic encase is used to determine the RNA molecules, and then we can find out the messenger RNA by using this DNA microarray technology. And discovery of the drug and leading compound. So discovery by using the different application of the microarray technology, we can find out the treatment. As far as individual personalized treatment is concerned, we can find out the expression profiling of the drug and microbial diagnostic of the drug, infectious disease and detection of the drug, as well as non-infectious disease and its detection and identification. So it will help for the proper treatment for the proper patient. Again, there is a personalized treatment where the genetics play the role. So genetic medicine also hold to promising predicting the disease, as well as such genetic medicine offer immense potential in the personalized treatment for the individual genetic makeup in the field of oncology also. It's a start from the oncology detection. Oncology treatment gives the, we can say, it's a target for the personalized medicine. It gives the clue for the personalized medicine. So an individual genetic makeup can be possible by find out the mutation in the cell, and then we can prescribe more effective drug and avoid the unwanted prescribed drug, which may cause the predictable side effect. So by increasing the patient adherence to the treatment by targeting the right patient with the right drug is the aim. So by using different technology, we can reach to the personalized treatment of the drug. And right now the targeting is also one kind of personalized treatment. So the dream of the Paul Enrich, he gives the bullet, magic bullet concept. So in the novel drug delivery system, there is magic bullet is also very added to the personalized medication system, where we can target the cancerous cell immediately by using the nano carrier system. So by reducing the time and cost and failure rate of the pharmaceutical and clinical trial, such personalized medicine can be used by using the different tool of the biotechnology and other technological aspect as far as concerned with the targeting nano carrier system. So to control the healthcare cost by avoiding unnecessarily cost where the drug is proven ineffective, the personalized medicine can be used. So in a nutshell, personalized medicine is very safe, but there is a lot of intervene of the technology, nanotechnology and targeted drug technology. So once the targeted technology is refined in a refined way, if we use, then personalized medicine or personalized treatment can be achieved with the better therapeutic effects. So this is in a nutshell about the personalized medicine, and I try to incorporate microarray system as far as biotechnological is concerned, but there is a different electronic and physics involved. Physics aspect is also involved in the personalized medicine, which I didn't mention in this presentation. So it's all about the personalized medicine and biotechnological aspect. Thank you very much.
Thank you, Professor Ashawat. So now the session is open for discussion. Among participants, if you have any questions or do you need any clarification from the speaker, please raise the questions here. So if there is no questions, I would like to thank again Professor Ashawat for giving such a wonderful lecture on biotechnology and personalized medicine. He introduced this topic by giving a familiarized disease, diphtheria and tetanus. He has given a brief introduction about it and how tailor-made medicine have started in 1980s. The example he has given is the invention of insulin to treat type 1 diabetes. And later in 21st century, the personalized medicine concept and the utility has increased at larger extent.
He has also elaborated on patient-centric care and customizing individualistic diagnostic approaches. And he has also introduced when the personalized medicine has initiated, that was initiated in 2015, and it has changed, it has provided a major role in the shifting paradigm of medical diagnostics. And he has elaborated on the use of diagnostic tools and as well as genetic medicine in personalized medicine. With this, I would like to thank again Professor Ashawat for your wonderful lecture. Thank you so much, sir.
Thank you, sir. Now, I'd like to invite Dr. G. Priya to introduce the chief guest of the third session.
Good morning to all. We are honored to introduce Professor Kapil Kumar, an esteemed professor and director at Samrat Prithviraj Chauhan College of Pharmacy, part of Uttarakhand Technical University in Kashipur. He brings over 17 years of rich research and teaching experience in the field of pharmaceutics. Sir has made significant contributions to the scientific society, publishing more than 105 original research articles and reviews in highly reputed international journals, achieving notable academic metrics with 1,356 citations. His recent publications reflect his extensive research, including studies on magneto-dermoelastic interactions, advancement in breast cancer treatment, and innovative chemical reductions in biofuels and life cells. His academic journey includes roles of assistant professor at Vivek College of Technical Education, associate professor at Global Institute of Pharmaceutical Education and Research in Kashipur, and professor and head of the Division of Pharmaceutics at the same institution. Additionally, he serves as the editor-in-chief of Universal Journal of Pharmaceutical Research. Currently, Sir continues his impactful work as director at Samrat Prithviraj Chauhan College of Pharmacy, furthering research and education in pharmaceutics. Please join me in extending a warm welcome to Professor Kapil Kumar, sir. Welcome you, sir.
Hello. Good morning to all organizers, all participants. First of all, I would like to express my thanks to Dr. Suresh, Dr. Mahadeva for the invitation of this event and hello, I am unable to share my screen. Please enable sharing of the screen.
Yes, sir, we are doing it. Yes, sir, add the screen. Yes. So now you may share the screen, sir.
Okay. Yeah. It is visible to everyone.
Now it's visible, sir. Yes, screen is visible.
Okay. And the topic is role of pharmaceutical sciences in addressing global health challenges. Basically, I would like to start my talk with the term that is health. Health in Indian tradition is the wealth. If health is okay, it means everything is okay. We can pursue the different thing because of good health. And a sick person, a diseased person, have some limitation, and it is even not supportive for anyone. The main is the pharmaceutical sciences, or another name is the pharmacy. It is the word derived from the Greek word pharmakon, meaning the medicine or drug. Basically, the pharmaceutical sciences is related with the collection, identification, synthesis, purification, isolation, quality control, medical substances. Basically, drug is the substance that is natural origin, that is maybe prepared synthetic, and that is used to control, treat, mitigate the diseases. And it is the art and science of preparing and dispensing drug. Now, our topic that is related to the health, according to WHO, health is a state of complete physical, mental, social well-being, not merely an absence of disease and infirmity. This is the definition of health, and there is a term that is international health. It is refers to interlocking and interrelated health status of people throughout the world and effort to improve the health of all people of every country. It is related with the all countries. It means the international level that there are attached with each other. The literature of one country is going to be circulated through the another country. There is the one more term that is global health. Health problem, issues, and concern that transcend national boundaries, which may be influenced by circumstances or experienced in other countries, and which are best addressed by cooperative action and solution. Meant to see, suppose there was a COVID in some years ago, and the whole world was affected. The global health of each and every country people were affected. No, this is the global healthcare. It is understanding of healthcare is an international and interdisciplinary context, includes the study, research, and practice of medicine with a focus on improving health and healthcare quality and population worldwide. Now, the objective global health. What is the need of the global healthcare? A high life expectancy and being populations, an expansive health and medical care system, a rapidly growing private health market. Health is a dominant theme in social and political discourse. And health is a major personal goal in life. Now, come to challenges in global healthcare. There are so many challenges that we have to each and every country is facing. There are the non-communicable diseases, global pandemic, fragile and vulnerable setting, environmental problems, including air pollution, climate changes, animal health, food sourcing and supply, economic disparities, and access to healthcare, antimicrobial resistance. These are the different challenges the whole world countries are facing. Now, non-communicable diseases. Diabetes, cancer, heart diseases, these are the most common diseases that are non-communicable disease. It is the person who are having the age 14 plus that are having these diseases. It is a very common factor. Approx in India, I say there are approx 80% people are having diabetes that are having more than 40 years age. Major reason to rise of these diseases are tobacco use, physical inactivity, harmful use of alcohol, unhealthy diets, and air pollution. Basically, these are some diseases that are related with the habit. And now come to the pandemic. The whole world is knowing about the COVID 2019. It affects the whole world, and the pandemic are global disease outbreaks. Examples are the SARS, Ebola, COVID, and other viral threats. Every year, there are newly emerging pandemic threats that cannot be solved, and diagnostic symptoms may only occur after an individual is already infected. In the latest report that was taken on 19 May 2024, 2 days back, approximately 70 lakh 47,741 people were died because of this COVID. And that was a major issue for the scientists of all countries. They were involved in the development of the vaccine, development of the medicine because of this COVID. And there are some other reasons. There are fragile and vulnerable setting, drought, famine, conflict, population displacement, all these factors that affect the health issues. More than 1.6 million people, 20%, 2% global population live in places where prostrated with crisis. There are not so healthy sanitation is there that is affecting the health of the people. And next one is the environmental problems. Growing concern about the environment because it affects the health of the public on climate change and air pollution. But how will these challenges directly affect the health of human population? In most cases, the answer lies in water sources and its sanitation. Main is the sanitation, how the people are living that affect ultimately their health. Air pollution is also the factors that leads to health issues. In 2019, air pollution, the greatest environmental risk was there. 9 out of 10 people breathe polluted air every day. Burning fossil fuels are the other reason. Now, come to the key concept in relation to global health. The determinants of health, the measure of health status, the importance of cultural health, the global burden of disease, the key risk factors for various health problems, the organization and function of health system. These are the key concepts that are related with the global health. Now, the determinants of health, genetic makeup, age, gender, lifestyle choices, community influences, income status, geographic location, culture, environmental factors, work condition, education, access to health services. These are the different other factors. Malnutrition is one of the reasons for the poor health. Cooking with the wood and coals that leads to the development of leads to lung diseases, poor sanitation, more intestinal infections are there. Suppose someone is living in the condition that are having the poor sanitation, poor life circumstances, just like the HIV/AIDS. These are because of the poor life circumstances. Advertising tobacco and alcohol. People who are using all these products will ultimately deteriorate their health. Rapid growth in their vehicle traffic, often with untrained drivers, so unsafe. So these are related with the traffic accidents. No, plus more general factors such as environmental degradation, population growth pressure, urbanization, development country precedents. Next is the measurement of health issues status. Cause of death often finds death certification, but limited because of incomplete coverage. Life expectancy of the birth. Basically, I would like to say one term, that is, if a lady is giving birth to a child, that is basically that second birth to that ladies. In previous time, there was no proper facility in approx all countries, and the ladies, there were birth death rate at the time of delivery was but noted in each and every country. The average number of years a newborn's baby could expect to live if current trends in the mortality were to continue for the rest of the newborn's life. It was not safe to the lady, mother, and as far as for the baby, because of the lack of medicine, because of the lack of proper facilities. Culture, the predominating attitudes and behavior that characterize the functioning of a group of organization, traditional health system, psychos, these all are factors that are related with the health. The global burden of disease predicted change in burden of disease from communicable to non-communicable between 2000. As far as the scientists concerned, what will be the reason of the death in approx up to 2030? There are so many reasons that related with the diabetes, that are related with the cancer, that are related with the heart attack, that related with the stroke. All these factors will least related to the death rate in the upcoming years. Key risk factors, the same is there: tobacco use, poor sanitation, malnutrition, are the reason for that. That is affecting the health issue. Now, come to this point, pharmaceutical science. As I told, that it is basically profession that is related with the medicine. The whole world people that are using the medicine all are covered by the professional that are related with the pharmaceutical sciences. The world deals with the pharmacy profession, with healthcare, and its main purpose to safe and effective use of pharmaceutical product. The medicine that are used, that are prepared, that are dispensed, that are marketed, all these people are the pharmaceutical science professional that are the pharmacists. Now, what is the main role of the pharmaceutical sciences person, that are the pharmacists? Develop new medicines, develop medicine, supply medicine, provide advice about medicine, offer health services. Pharmacists also work for the awareness in the university regulation, government organization, research publishing, and the pharmaceutical industries. The different areas for the pharmaceutical person, I think in this conference, mostly people are of the biotechnology group. So these are the different fields in which pharmaceutical person work: pharmaceutical marketing, regulatory bodies, they can serve as scientists, abroad research, they are the different prospects, academic, in production, manufacturing, formulation, development, hospital, in the hospital, they can serve with the pharmacist, community pharmacist. So these are the different ways in which a pharmaceutical person, a pharmacist, serve the society for the good health. Now, I would like to give the brief description in the form of community pharmacy. It is retail pharmacy. Any person who is graduated, who has the diploma in the pharmacy, can serve as a retail, can open a retail shop and can serve the society by providing the medicine. Another form is the hospital pharmacist, the person that is present in the hospital that used to deliver, provide the medicine to different patients visiting to the hospitals. And the main purpose of the hospital pharmacy is compounding medication for use in the hospitals, helping patients to understand their medication and how to take them. Basically, that he gives OC give the instruction to the patient that are coming to the hospital, conducting clinical trials to cover new or modified treatments for rare diseases, and assisting specialized medical care such as cancer patient. No, industrial pharmacy related with the work of a pharmaceutical science in the industry, that is related with the manufacturing, that is related with the research, that is related with the quality control, that is related with the marketing, that is related with the dispensing of the medicine. The main purpose, the main work of pharmaceutical science of pharmacists are the marketing, legal department, sales, professional communications, clinical research scientists, research and development, production. So, as I told previously, that all medicine that we are using in our home, that are the work of a pharmaceutical person, that are work of a pharmacist in the different form. Manufacturing is the preparation of different dosage form, just like in the time of COVID, all scientists were working for the development of the medicine. The pharmacists were also engaged, the whole pharmaceutical industries were working for the trials, different preclinical trials. Another thing is the research and development. If a drug prepared in the market, that is already existing in the market, there is need to develop, there is need to make it more effective. So different scientists are working regularly, that are the pharma people, that are pharmaceutical sciences people. And the process of drug discovery, development is lengthy, complex and expensive. But still, there are so many big pharmaceutical companies that are engaged themselves for the development for the new drug. I mean, they are improving the quality of the existing drug. That is the main work. And another is the distribution. Distribution means how the medicine are circulated. If a country that is well developed, that is supporting to another one. A state that is well developed, that has the manufacturing plant, providing medicine to the other other states. In the time of COVID, India supplied vaccine to 160 countries. That is an impressive thing that the government of India has done during the COVID time. That serve as an elder brother for the different countries and for the neighbor one. And these are the clinical trials, basically, that are performed by the pharmaceutical person, by the medical person, and by the different team of the medicine profession that used to animals, that is preclinical study. Clinical means that use the human beings for the perfectness of the drug, for the checking of the therapeutic effect of the drug, for the checking of the possible toxic effect of the drug. Another is the advocacy and education, that is raise awareness about disease and treatment, and patient group, healthcare professional, policymaker, basically the pharmaceutical company, the pharmaceutical sciences involve themselves for the education, for the disease, and for the development of the different new medicines. No, how pharmaceuticals are used to treat and manage global health cases. What is the role? So apart from in the different ways I have discussed here, pupils are used, the scientists are used that are engaging themselves for the development of the new drug, for the development of different formulation, for the society. Pharmaceutical play crucial role in the treatment and management of various global health issues. In the case of HIV/AIDS, antiretroviral therapy has transformed the disease from a death sentence to a manageable chronic condition. Means if this medicine are provided to the patient, that can be manageable chronic. That there will be no drug, there will be some circumstances that can change to the manageable chronic condition. Similarly, medication of malaria and tuberculosis have been vital in reducing global burden of infection disease. Beyond infection, disease and pharmaceuticals also indispensable, such as cardiovascular disease or pathology that could lead to them from such as MASLD and mental health disorder. Medications like statin and antidepressants have significantly improved outcomes for millions of individuals worldwide. Basically, I would like to add one more point. That is, in these days in which we all are living, we are having this many stress in our life. That there are so many diseases that are because of our lifestyle, because of not because of that are communicable, because of that how quality foods we are eating, that is affecting which environment we are living. So medicines are the essential thing. The medicine means there is involvement of the pharmaceutical sciences, involvement of the pharmaceutical persons. And understanding the role of pharmaceutical disease prevention, not only treat but also help prevent disease. Vaccine, for instance, have been instrumental in eradicating or controlling disease for polio and measles. And I would like to recall once again the COVID. At the time, whenever the COVID was going on, we were searching in the newspaper whenever there will be the new vaccine, whenever there will be new medicine for the treatment of that COVID. In addition, medications such as antimalarials and prophylactic antibiotics plays a pivotal role in preventing infection and reducing the spread of disease. Disease can be controlled, that can be mediated, and even we can prevent the spreading of the medicine. There are so many diseases at the time that are fully eradicated from the society that are not at present. Suppose the polio and measles. Yes, there are so many cases, but still that the cases are very less. As far as in the previous time there was concerned, there was so many cases of the polios, and but at the present time there are very few cases because of the government policies, because of the pharmaceutical involvement of the pharmaceutical persons. In conclusion, I would like to say the role of pharmaceuticals in global health is undeniable. They have revolutionized medical treatment, eradicated diseases just like the polio, and improved patient outcomes worldwide. So, thank you everyone for listening me, and thank you for the organizer. Thank you giving me this opportunity. If there is any question, you may please ask to me.
Thank you, sir. Participants, if you have any clarification, you may ask, sir. If not, you can post your questions in the chat box also. Yes. If no more questions, thank you, sir. Thank you for shedding lights upon its challenges, also communicable and non-communicable diseases and its pandemic situations across the global. And mostly during the COVID, which you have taken as an example, to share your thoughts, and also growing concern about the environmental problem with respect to healthcare, and the role of pharmacists as well as the pharmaceutical industries in manufacturing as well as in research and clinic development and distribution across the globe. So thank you for sharing your insights about the pharmaceutical sciences and global sciences. Thank you, sir.
Okay, sir.
Now I invite Dr. G. Priya ma'am to give the introduction about the fourth session, Talk Resource Person. Priya ma'am, you may proceed.
We are pleased to welcome Dr. Ahmed Najib, esteemed Associate Professor of Natural Product Chemistry at the Faculty of Pharmacy, Universitas Muslim Indonesia. Also known as Najib, he brings extensive expertise in pharmacognosy and phytochemistry. Professor Najib Sir has been a dedicated educator and researcher since 2004 at Universitas Muslim Indonesia in Makassar, South Philips. He has also served as head of the Laboratory of Pharmacognosy, Phytochemistry from the year of 2011 to 2019 and continues to contribute significantly as a senior lecturer. His academic contributions extend beyond teaching. He serves as a reviewer for journal Mandela Pharmacon Indonesia and as a member of editorial board of Universal Journal of Pharmaceutical Research. Professor Sir research and scholarly work includes over 135 publications with recent notable contributions such as evaluating the free radical scavenging properties of compounds isolated from Syngium oblanceolatum, exploring new bis indole alkaloids and comparative studies on the antioxidant activities of turmeric and curcuma extracts. His work has been published in reputable journals such as Chemical and Pharmaceutical Bulletin and AIP conference proceedings, etc. Please join me in extending a warm welcome to Ahmed Nazib, sir. Sir, welcome you, sir.
Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh and good afternoon, everyone. So I'd like to thank to all of committee, including SRM Institute of Science, Faculty of Science of Humanity, and Department of Biotechnology, University of Zainal Abidin, Sultan Zainal Abidin, Malaysia, and also Laureate Institute of Pharmacy, Chemical, Pradesh Technical University, India. So this is a great change to share my work. So please, I will share my presentation. It's visible? Okay.
Yes, sir.
Okay.
Yes, sir.
In the last session, I will talk about the Cordia mixa plant, the potential for the anti-diabetic. As we know, there are 7 layout and introduction on in this presentation. So the first introduction, the second one is a botanical description on this plant from traditional use, phytochemical constituents, anti-diabetic potential, conclusion, and annulment. As we know, Indonesia is a second mega biodiversity country after Brazil, and it's covered to 15.5% of the world flora. So Indonesia have traditional, like India, there are traditional medication as jamu. There are on the Sulawesi Island, the Celebes Island, is the 4th largest island in Indonesia. It's located in Australia zone and Wallace Island. This is high number of medical plants. So the poorly studied are in Southeast Asia. So the South Celebes especially still more potential to explore for the plant medicine. So the unique novel and bioactive compounds was found on from this area, and the need to investigate for the phytochemical study. On this our research, and I will report our research results from Kanunang, the local name from Enrekang, and that's take from June 2018. This is part of plant of the Kanunang. The scientific name is Cordia mixa, and also names Asiram plants, Lasura, Sebastian plant from familia Borgonaceae, and distributes are native in Asia and also Africa, and widely cultivated in tropical and subtropical region. The key characteristic is medium size and deciduous tree up to 50 meters tall, and broad opaque rock texture from leaves and flowers small, white to pale yellow and fragrance. And the fruits is oval shape, drupes green to yellow or brown when it ripens. The medical respiratory use from traditional use to use cure the gastrointestinal disorder and also skin disease, and fruit as a culinary, eaten as fresh and used as pickles, jams, or sauce. There are some reports to this plant as a hepatoprotective activity from the methanol extract, and it can show the carbon tetrachloride induced lipidemic and thiocyanate or TA. And also report as antioxidant activity that the methanol extract of fruit and leaves was reported to show antioxidant activity in DTPH radical scavenging method. It's reported by AFSA and in 2007. There are many compounds have been found in the Cordia genus itself. For example, is the marantine tryptophan from the Brazilian medical plant Cordia mustifasciata, and the cytotoxic monoterpene for benzoquinone and root of Cordia globulosa, and then there are glutamate alkaloid and terpenoids, benzocaine from Cordia globifera. So on this plant, or especially from the genus of the Cordia, find many substances, and then just antifungal and herbicidal compound from the root bark, Cordia aliodora, and for example, anti-leishmanial compounds from Cordia Prognostima, colleague in Burma and Burma, and and biomimetic transformation from the biological activity of globiferin as find a terpenoid benzocaine from Cordia globifera. The interesting one that we have isolates come from enhexan fraction, especially from Cordia mixa L. From this compound one, we find octason and stigmasterol and decadionic acid or methyl ester, and there are more 9 compounds have been isolated from enhexan fraction, such as cinnamon acid until pomethiol. Interestingly, even all of the compound is known, but for the compound 5, for hydroxymethoxymethyl benzaldehyde, this is the first compound but isolate from genus of Cordia. And for compound 5, 4 and 9 is the non-compound, but this is the first isolate from Cordia mixa, cinnamic acid for hydroxymethoxymethyl benzaldehyde and pomethiol. As we know, the mechanism of glucose blood regulation in our body is start by stomach, and then belong to bloodstream on our intestine, and then make a mechanism of pancreas to release insulin, and then the liver will be a storage glucose, and the body cell will be storage glucose. If the mechanism is not longer established, the human or people will be get diabetes. So there are many plans to cure the diabetes. For example, the mechanism to increase glucose production from berberine, fenugreek leaves, and also to increase glucose absorption for example, Mircea and Sangsi. Then the mechanism, as the third mechanism, is increasing the insulin secretion. For the example, to plant ginseng, bitter melon, aloe, and Rotopilum sensitivum. And then the last one is the increase the peripheral glucose uptake, such as consume ginseng, bitter melon, and cinnamon. On this our research, we use in vitro method to prove that the potential action of Cordia mixa. In the charts, you can sample after fractionation, and then we assay with ELISA using the in vitro method by the enzymatic system. Then we find the result that enhexan extract have activity to inhibit alpha glucosidase with IC50 value 0.53 ppm. Now, the research said that all of fraction 1 until fraction 9 have a potential to inhibit activity of alpha glucosidase activity. So, for the first fraction is the most potent to inhibit the activity of alpha glucosidase. And then if we see the reaction for the mechanism of line work analysis from the kinetic enzyme that we find the compound in extract is work as mechanism of non-competitive. So it will be approved between the component of this extract, the way of they inhibit of alpha glucosidase. In the in silico method, we find that all of compound on this plant is have a mechanism to inhibit alpha glucosidase. The all compound is found from GCMS analysis and then docking program using Autodoc, and we find that 19 of compound have average energy that have inhibit alpha glucosidase, and the result that the compound of 2,2,4 trimethyl 3,3,8 2,16 tetramethyl heptadeca 3,7,11 15 tetradecyl cyclohexane have delta G or free energy minus 9.3 kilocalorie per mole, and also in the bis 2 ethyloxyphthalate have free energy minus 7.8 kilocalorie and kilocalorie per mole. So the 19 chemical compounds of protein mixa have sorry, value of the free bonding energy delta G in the range minus 5.3 kilocalorie per mole to minus 9.3 kilocalorie per mole, and two compound with the higher delta G followed than the others that are bis 2 ethylene ethyl phthalate and 2,2,4 trimethyl 3,3,8 2,16 tetramethyl heptadeca 3,7,11 15 tetraenyl cycloxyanol with the delta G minus 9.3. On in vivo experiment, we find that the Cordia mixa have a potential as an anti-diabetic in mechanism of alpha glucosidase inhibition, and we find the potential is 76.44 and 12.15 and 63.64. This is show that the Cordia mixa has potential as antidiabetic agent due to its phytochemical profile and multiple mechanism of action. Our further research need to establish or standardize a dosage and confirm efficacy. So we will welcome to collaboration with our group, and we will develop this plan in the future. So we will enrollment to Director of Research Technology and Higher Education, Ministry of Research, Technology and Higher Education, Republic Indonesia for funding this research. And then also gratitude to Associate Professor Kyoko Nakagawa-Goto from the Laboratory of Natural Products and Medicinal Chemistry at Kanazawa University, and then to facilitate us for identification of a Cordia mixa compound, and then also grade 1 from our faculty member. So we will collaborate on this future. So thank you very much. Hello?
Hello. Thank you, sir. Thank you for sharing your insights about the plant. This is plant Cordia mixa and its antibiotic potentials. and also the phytocompounds, especially having a major role in antidiabetic properties. And the research you have shared has produced some insightful considerations about the antidiabetic potential of the plant. Thank you, sir. If participants, anybody have any clarification or if you need to ask anything, you may kindly come up with your queries. Participants?
Hello, am I audible?
Yes, sir.
A very good morning, sir. Thank you for such a nice presentation. Sir, I wanted to know that as you have done in silico study by utilizing.
Okay. I answer the question, Dr. Jennifer.
Hello.
My answer the confirmation from Mr.
Is having these kind of effects.
This is actually our research using three methods. For example, such as in vivo, in vitro, and also in silico. On in silico, we found the compounds using GCMS, and then the results of the compounds from Codemixa. It was docking by the program Autodoc, and then show in my presentation. There are 19 compounds, and only 2 compounds have a high potential than the others. Mr. Misra?
Yeah, sir, actually, I need to know, is there any genetic technique in silico genetic technique which can be utilized to find out this potency?
Okay. First of all, because for the in vivo and in vitro results using the alpha glucosidase enzyme, so we use a docking method by the alpha glucosidase enzyme for the PDB code 1LWG, and then after docking, and then evaluate the free energy, and we find the results as I told in my presentation. So for genetic, there are we don't have research from the genetic, but in the future we will continue in this research by the genetic approach using the fruit fly, yeah, and then using a mechanism of the anti-diabetic rats with induce for the alpha glucosidase mechanism.
Okay, thank you very much, sir.
Okay, thank you, Mr. Misra.
Any other participants? You need any clarification, you may come out with your clarification questions. If there is no more further clarifications, thank you, sir. Now I invite Dr. Vinod Kumar, sir, to come away with the concluding remarks for the international seminar. Welcome, sir.
So very good morning to all the dignitaries and participants. It's such a wonderful session we have all seen since morning. So it's my pleasure to give concluding remark for this one-day international seminar, which is being organized by the Department of Biotechnology, Faculty of Science and Humanities, SRM Institute of Science and Technology, Ramapuram, Tamil Nadu, India, in association with Faculty of Medicine, UNISA, Malaysia, and Laureate Institute of Pharmacy, Himachal Pradesh, India. So this one-day international seminar on biotechnological advancements in healthcare was formally started with the inaugural session by our today chief guest, Professor Harmi bin Mohammed Yusuf, who has elaborated on the different biotechnological tools that is being used for health sciences which is followed by 4 different sessions. Session 1, the lecture was delivered by Dr. Saravalan Dharmaraj, Associate Professor, Faculty of Medicine, UNISA, Malaysia. He has delivered his lecture on functional analysis of obesity-related genes, selected by chemometric analysis of gene expression data from mice-fed high-fat diet. Followed by the second session, the lecture was delivered by Professor Mahendra Singh Ashawat. He is currently the Director and Principal, Laureate Institute of Pharmacy. And the third lecture was delivered by Professor Dr. Kapil Kumar, Director, Samrat Prithviraj Chauhan College of Pharmacy, Uttarakhand Technical University, India. The last session, the lecture was delivered by Dr. Ahmad Najib, Associate Professor, Natural Product Chemistry, Faculty of Pharmacy, Universitas Muslim, Indonesia. All the sessions were warmly attended by faculties, researchers, and students, undergraduate, postgraduate students from India, Malaysia, Indonesia, and other countries. We have more than 100 participants who are actively participating in such a wonderful international seminar, which is happening since morning. So, with these, in the end, I would like to thank all the people who are involved in organizing such a one-day international seminar. First of all, I would like to thank Chairman, Co-Chairman of SRM Institute of Science and Technology, Ramapuram, who is guiding us to organize such a event like this, which will help researchers and undergraduate, postgraduate students to update and develop their skills. And apart from them, I would like to thank Co-Chairman, Co-Director, Chief Director, Associate Director, and the Dean Academic and Dean Research for their valuable guidance. And importantly, I would like to give my heartful thanks to our Dean, Major Dr. Venkat Ramanan sir, for his guiding support. And also, I'd like to extend my gratitude to our Vice Principals, Administration, and Academic for their encouragement to organize such an event. And also, I would like to extend my gratitude to our head of the department for constantly encouraging and guiding us to organize this particular event. And more importantly, on behalf of the convener and co-convener, I would like to thank all our department faculty members and as well as the student coordinators who has helped to complete this international seminar in a high note. And at last, I would like to thank all the technical peoples supporting and non-teaching and hospitality people for organizing this wonderful event today. So thank you so much. And at last, I would like to thank all the participants who are joined from various institutes in India as well as from other countries. And also requesting all the participants to encourage us and support our department in attending all the events which is being organized in future. Thank you so much.
Dear participants, thank you all for your enthusiastic participation from the beginning till now. And the feedback link is shared in the chat box. The link will be also shared in the WhatsApp chat group. So thank you all. Now let's conclude the session with the national anthem. Dear participants, once again, thank you all for your enthusiastic participation. Thank you all and have a nice day. Thank you.